Streaking colonies. Streaking is a technique used to isolate a pure strain from a single species of bacteria. This is done by isolating single colonies. A colony is a collection of bacteria resulting from a single bacterium and so share the same genetic characteristics. For making a pure culture streak, you will need sterile inoculation loops. Watch the video about sterilizing an inoculation loop to see how this is achieved. It is important to allow the loop to cool down completely. This prevents the bacteria from being killed by the heat. Look at the original plate and find a colony that is isolated and not in contact with any other colonies. Before staining, label an empty plate that you will use clearly. Label the base of the plate with your name, class, test, date, and kind of bacteria. If necessary, also note the incubation conditions. Work near a blue flame in order to prevent any contamination. Place the plates with the lid facing the table. This enables you to take the plate using only one hand. For right-handed people, take the plate with a grown bacterial culture in your left hand and an inoculation loop in your right hand. Take only a small amount of bacteria, just touching the colony is enough. Now place the plate back and put it aside. This plate with the bacteria is not needed anymore. Take the coded empty plate with your left hand and spread the bacteria over approximately 1/4 of the sterile plate. You do this by drawing the inoculation loop back and forth on the growth medium. After streaking the first sector, flame sterilize your loop. To obtain isolated colonies, we have to distribute the bacteria from sector 1. Use the cooled sterilized loop to streak the second sector. Drag the loop about 3 times through sector 1 and make 3 additional individual streaks. Use the other side of the inoculation loop to streak the next sector. After all, on this side of the loop there are no bacteria present. Again, drag the loop through the previous sector a few times, followed by approximately 3 individual streaks. Now flame sterilize your loop. Use a cooled and sterilized loop to streak sector 4 of the plate by dragging the loop through sector 3, followed by individual streaks. Use the other side of the inoculation loop to streak the next sector. To streak sector 5, drag the loop through sector 4 and then continue in a zigzag pattern towards the middle of the plate. Be careful not to touch any other previously streaked sectors. Sterilize the loop when finished. Close the plate and turn your Bunsen burner off. See under what conditions the plate should be incubated. Label a rack for your plate and put your plate upside down in the rack. Put the rack in the incubator. The streak plate is successful when there are isolated colonies in the 3rd or 4th sectors and only one type of bacteria is growing on the plate.